Welcome to the ChinaBio Partnering event. I'm Brian Yang, the senior editor with Scrip Pharma Intelligence. With me today is Dr. Alan Lu. Dr. Lu is senior vice president for Sirnaomics. And welcome to the interview, Dr. Lu.
Thank you, Brian, for inviting me.
Could you introduce your company to us, please?
Sure. Let me spend 1 minute to introduce myself and then my company. I finished my college education in China. I graduated from University of Science and Technology of China with BS degree. Then go to Shanghai to finish my master's degree in Shanghai Institute of Biochemistry. Then I went to US to finish my PhD in University of Illinois at Urbana-Champaign. I spent about 8 years as a faculty in Johns Hopkins. But after I got my MBA, I left Johns Hopkins to join the industry. Sirnaomics is established in 2007 in Gaithersburg, Maryland. Sirnaomics is focused to develop new therapeutic drugs with the new technology called siRNA interference. So our company, the name is siRNA, small interference RNA plus omics. So that means we use small interference RNA, but we combined several different ones. We use omics idea.
Dr. Lu, can you give us some ideas how your company has progressed since the establishment?
Okay. siRNA therapeutic area in general is started from the end of last century when the two US scientists Craig Mello and Andrew Fire published the seminal paper in Nature in 1998 to show RNA interference in mammalian cells. Pharma companies realized this technology can be applied to develop drugs. RNAi or RNA interference is this process. We all know our genetic information is stored in DNA. DNA is transcribed to messenger RNA. Messenger RNA is a mediate or is the middle step to translation into protein, all the functional molecules. RNA interference is a small piece of RNA which can base pair to the messenger RNA, recognize the messenger RNA and degraded the messenger RNA. So when the messenger RNA copy number is decreased, the protein number translated will be decreased. If this messenger RNA and this protein is the protein related with the disease will be cured in this way. So our company is established to develop new drugs pure by applying RNA interference technology. But in recent years, we also explored to microRNA and messenger RNA areas. In 2008, our innovation team came back to China. We established Suzhou Siraonomics in Bio Bay. Suzhou Siraonomics is a kind of extension of the US Siraonomics. If we want to divide the function of these two parts, I can say the US Siraonomics is focused on R in R&D, and the Suzhou Siraonomics is focused on D of R&D. In 2012, our team also established Guangzhou Nanotechs to finish the pilot production formulation to prepare for the clinical trials.
Can you give us some introduction about your pipeline and what are the therapeutic areas that you are focusing on?
Sirnaomics is a platform company. The platform is composed of the following major parts. The first part is a delivery platform. We all know if you want to achieve the purpose to interfere the messenger RNA functions, you have to introduce small interfering RNA into the cells. However, the cell membrane is a hydrophobic barrier, just like bubbles, just like oil bubbles. It's not easy for a hydrophilic cell to cross the membrane. So if you want to cross the membrane, you have to have a delivery system to pack the small interfering RNA, just like a small bubble. When they meet the cells, they can merge with the cells and release the siRNA into the cells. So the first major technology is the delivery system. We have 3 generations of delivery systems. For the first generation, the most one we're using now is a peptide we call histidine lysine polymer, or in short name HKP. HKP is invented by Professor James Mixon in University of Maryland. HKP is composed of mainly the positive amino acid, histidine and lysine. In neutral pH, those amino acids are positive charged, but siRNA or DNA or any nucleotide are negative charged. When you put these 2 molecules in solution in neutral pH, they can form nanoparticle just by nature. So the nanoparticle is very stable. And in our hand, we can prepare the nanoparticles in dry powder, and it can be stable for up to 24 months. So this is the first platform. The second one, we have from a design to validation in vitro, in vivo, in animal, the whole system. For example, if a new virus is discovered, like a Zika virus was discovered from South America, then the virus sequence is determined. We can quickly find the sequence and design the siRNA to against the sequence. Then we validate in cell line, in animal, and validate if this sequence can be a good active pharmaceutical ingredient or API. So we can develop the drug very quickly. The platform is mainly composed with these 2 parts. For the other parts, I can just omit it.
You told during your presentation that you are going to enter the clinical trials for one of your assets. What is the process? Where are you going to develop the asset?
Our product can be divided in 3 major areas. One is anti-fibrosis. The second one is anti-cancer. The third one is antiviral infection. And now our first leading product in anti-fibrosis area is under clinical phase 2 trials in the US market. And we just got FDA approval for the Chinese clinical trials. So we are going to start the Chinese clinical trials pretty soon. The first clinical indication for this drug is for a skin hypertrophic scar treatment. However, because this drug is designed to target both TGF-beta1 and COX-2, 2 targets, this can be used for general anti-fibrosis effect. For this drug, the second medical indication is a cholangiocarcinoma, is a liver cancer. And the third medical indication is PSC, primary sclerosing cholangitis. And the fourth clinical application will be a non-melanoma skin cancer. For the second and the third medical indication, we're going to file IND in the US this year for orphan drug, for orphan indications.
What are some advantages for a US company to develop a new drug like what you're doing in China right now?
For our company, we have a special setting. Our headquarters is in Gaithersburg, Maryland, but we have operation in China, in Suzhou and Guangzhou. In our experience, we can take advantage in both sides. For example, we can use the Chinese animal facilities, relatively lower human power, to finish all the related animal safety studies and the enabling studies. At the same time, because the CRO we are using has the ability to file IND in both in China and in the US. So their experiments can support the IND filing both in China and in the US. We use the same experiments to support our IND in China and in the US. For the US FDA, because IND processing is very quick. For example, we filed the Chinese IND in October 2014. We just got the Chinese CFDA approval in March 2017, almost 30 months. But in the US, we filed the IND in October 2016. We got approval in November 2016, only 1 month. One is 30 months, the other one is 30 days.
When you come to this partnering event, what are you looking for?
For our company, we are a small biotech company. Even though we have technology advanced platforms, we have we believe a very good product to meet the unmet medical needs all over the world. However, we still need to find, for example, big pharmas. Big pharmas, this is kind of like a marriage. The big pharma is always looking for good technology, good products from a small biotech. And small biotech is the hunger of fund. We need the resources. We need the resources, not only money, but the marketing capabilities and others. So I'm looking for see if big companies like Pfizer, Eli Lilly, Merck are interested in our technology and the products. At the same time, because we are in a Series C fundraising process, I'm also looking for some financial groups who is interested in our company and our technology.
Just very briefly, what's your financing situation now?
We finished our Series B finance in, we closed in 2015, in July 1st, 2015. And with this fund support, we pushed our clinical programs advanced very fast. That's why we can finish the US IND filing and start the Phase II clinical trials. By the way, the clinical trials now is going very well. We finished the Chinese clinical trials. We also push the other therapeutic products advanced very forward. Now we plan to close. We divide the C round in two parts in order to meet the Chinese RMB financial group needs and the US dollars financial group needs. So we divide this round in two parts. The C1 part is mainly for the Chinese RMB. It's $20 million equivalent RMB. We are going to close in middle of July. And for the second part, the US part, the $20 million we are going to close 3 months later.
Thank you very much, Dr. Lu.